Ready for the weekend? Nobody's ready for the weekend? I will register one. One is ready for the weekend. All right, so a couple of things. Number one, I had several people ask me about problem number 18 in chapter 1. How many of you have looked at problem number 18 in chapter 1 and it seems like, oh my God, right? What's going on with that thing, right? Okay? Well, problem number 18 is a prime example of the kind of thing that I'm talking about with respect to practical knowledge and practical chemistry and practical mathematics. You look at problem 18, and at first it seems like, oh, well, I can run through and I can do these calculations and everything is fine and dandy because now I know the Henderson-Hasselbalch equation, and then all of a sudden you hit a brick wall. Because you're obsessed with numbers. Because it asks you for a number, that's reasonable. All right? But what you discover is when you're doing problem 18 that Henderson-Hasselbalch equation doesn't work for you in the last half of the problem. That's a little unsettling, right? Now, I don't give you problem 18 to give you a whole new other set of things to learn about calculating pH. I give you problem 18 to make you think about something. I don't care if you get a number for the last half of problem 18. That's not the important point. The important point is understanding what's happening in that system. Let's think about this. You start with a buffer. In the first half of the problem, you work with a buffer that has a concentration of 0.1 molar, or 0.01, whatever it is, 0.1 molar, right? And you add HCl to that buffer, and the amount of HCl that you add is always less than the amount of buffer that you have. And the equation works great. And then when you get to the second half of the problem, you've got a much more dilute buffer, and you discover that you're adding as much or more HCl than you have of buffer. And the most natural thing in the world that should happen does happen. The equation falls apart. Okay? The equation falls apart. Why? Because buffers, and here's the take-home message of problem 18. If you want the answer to problem 18, here it is. Buffers have a limited capacity. If you exceed the capacity, you no longer have a buffer. Gone. Henderson-Hasselbalch, gone. Buffer doesn't exist under that condition. That's what problem 18 is about. It's about understanding what's happening in that solution and not about obsessing with numbers. Because every student I've talked to about it, and I've talked to numerous students about it, they're obsessed with the number. And I can't get them to think about what's happening in the solution. Okay? Why can't you get the number? What does it mean when you can't get the number? Well, it doesn't work. I can't get a negative logarithm. Well, what does that mean? The problem is you're focused on the number and not what's happening in the solution. That's a really good example of the kinds of things I'm talking about. Does that make sense? Do you feel better about problem 18 now? No? Okay. If you have problems with problem 18 or whatever, come see me. I'll be happy to meet with you. Okay. Let's get... What is this thing doing? It rearranges every time I try to do this. Yeah, we don't want to have all that. Come on, you. Oh my God, I gave away a secret there. Oh, no. All right, we'll work it over here. I'm not quite sure why this thing decides to rearrange my windows. All right, let's let's move now into a more practical application of the kinds of things we talked about with respect to pH, pKa, and charge, and talk about the building blocks of proteins. All right? So the building blocks of proteins, everybody knows, are amino acids. Amino acids, there are 20 that are what we call the most common building blocks of proteins. And most common meaning 99.999% of proteins. Right? We find occasional rare amino acids in proteins, and we'll talk briefly about those later, but for all practical purposes, 20 amino acids are what we find in proteins. Okay? I've talked about how structure determines function. All right? Structure determines function. Okay? This figure, which is not an ideal figure, I'm not quite sure why it's overlapped like that, but this figure gives an illustration of protein structure that we're going to talk about. We're going to talk about it at 4 different levels: primary, secondary, tertiary, and quaternary. Okay? Protein structure is important because protein structure determines function. All right? And this illustrates for us on the screen what primary, secondary, tertiary, and quaternary looks like. I'm going to briefly tell you what these are, and then I'm going to talk about them in more detail later. All right? Primary structure is the sequence of amino acids that occurs in a protein. Primary structure. Primary structure determines all of the other properties of a protein. The primary structure determines all the other properties. It determines the secondary structure, it determines what the tertiary structure is, and it determines what the quaternary structure is. If we change the primary structure of a protein, we change the sequence of amino acids of a protein, we will change the other components of a protein. We will change the secondary structure. We will change the tertiary structure. We will change the quaternary structure. Okay? That's very, very important. You can see illustrations of these, and we'll talk more about those. So I'm not going to go through those illustrations at this point. Okay. Here's a really cool example I like to talk about in terms of structure and function. All right? We'll talk later, actually next term, about this protein that you see here. The protein is the part in yellow and in red. Okay? This is a protein whose job it is to hold a DNA polymerase onto a DNA molecule. That's its job. It's called a beta clamp. All right? You don't need to know that right now. You can see the DNA that it holds onto is in the middle. And you can see the clamp is just like a clamp. The way it gets on there is it starts out like a little C, and then it's closed around the DNA. And when it's closed around the DNA can't go flying away. The protein is holding onto it. This protein holds onto a DNA polymerase, so the DNA polymerase doesn't go flying away. Simple structure, simple function. Okay? Structure and function are interrelated. Okay. I'd like to show this one. Okay. One of the things that we discover when we look at protein structure very carefully, particularly if we look at things like viruses, is that proteins, all right, can have the ability to self-assemble. A viral capsid has an ability to self-assemble. That is, to take, you could imagine taking a can full of pieces of a puzzle and shaking it up and throwing it out here on the thing, and the puzzle solving itself. That's really what self-assembly is about. Okay? Certain proteins have the ability to do that, and viruses long ago figured that out, so they can make a coat to put their nucleic acid into. When we think of nanotechnology and we think of things that work at that level, all right, proteins had that figured out long before there were human beings trying to engineer the same thing. Okay? Another thing we're going to talk about a lot is flexibility and function. Okay? This figure shows a protein that binds to an atom of iron. And when it binds to an atom of iron, you can see, as it shows on the screen, that the structure of that protein changes considerably. Same protein on the left and on the right. I'm missing my pointer. Same protein on the left and the right. The only difference is that the protein on the right has bound to the iron, and as a result of binding onto that iron, the protein's shape changed. The protein couldn't do that if it wasn't flexible. And flexibility turns out to be absolutely essential for protein function. Proteins are not rigid things. That'll become very, very obvious when we talk about enzymes in a bit. Okay? Enzymes do magical things, and the magic comes about because enzymes are flexible. Okay. Well, if we're going to talk about proteins, we have to start at the simplest level, and we're going to start by talking about amino acids. Okay? We're going to start by talking about amino acids. All right? Everybody learned in organic chemistry that if you have a molecule that has a carbon and if that carbon is bound to four different things, that there's two different ways that those four different things can be arranged around that carbon. Okay? Two different ways in which they can be arranged around that carbon. One is known as the L form, and one is known as the D form. Okay? First of all, I'm not going to ask you to draw a D or an L of an amino acid. Okay? I think you should know that there are two different forms. And the reason you should know there are two different forms is because when we look in biological systems, there's an amazing bias. Essentially, all of the amino acids that are found in proteins are in the L form. They are not in the D form. Why is that the case? Well, life evolved under conditions where having similar things made a lot of sense. What I use for my food is what you used to make the things that you did. If you used one form and I used a different, I can't eat you. And since we came from things that we ate, that was passed on and on and on and on. Another reason that happens is because proteins are made in very stereospecific ways. Having consistency in the ways, the arrangement of those amino acids in a protein allows the cell to have simplicity, not confusion. All right? There are minor exceptions to that rule, but they're very, very minor and they're interesting. All right? I won't talk about them at this time, but they're very, very minor and they're very interesting. Bacteria have them. Okay? But they're very rare. All right? Essentially, all the other proteins that are found as a result of being made by cells on Earth are in the L form. One of the things that astronomers and people who like meteorites and so forth do is they love to get a new meteorite that has fallen to Earth and look in it. And you can find amino acids and you can find various organic compounds in these meteorites. And the question that they ask is there a bias in the amino acids that are in there? Because if there is a bias, that would be a pretty interesting way of saying maybe that something biological made that. Okay? There's nothing magical whether it's D or L. On Earth, it's L. It could be D somewhere. Okay? But the question is there a bias? And answering that question is a very cool thing. So far, we haven't found meteorites that have that bias, but remember, we're a small portion of the universe. Okay. You guys have been seeing things about ionization and pH. I'm going to show you a figure in another way about ionization and pH. Okay? This figure, okay, shows an amino acid. Okay? And by the way, I should have pointed out, let me go back a figure. I'll show you, make sure I make a point about this. You're not going to draw amino acids. I will tell you what you need to know about structures of amino acids. One of them is right here. All right? Every amino acid that is found in proteins has the same general structure. It has something known as the alpha carbon. The alpha carbon is the carbon to which four things are attached. That's the asymmetric carbon of an amino acid. The alpha carbon is the asymmetric carbon. The four things that are attached are an amine group, a carboxyl group, an R group. You remember an R group in organic chemistry means could be a variety of things. It's a variable. And a hydrogen. Okay? That's the four things that we find attached to the alpha carbon in there. Okay? The carboxyl is called the alpha carboxyl. So we have a lot of alphas here. We have an alpha carbon, we have an alpha carboxyl, and the amine is called the alpha amine. Okay? So just some terminology for you there. You're not going to memorize structures, but you are going to need to know something about structure. I'll get into that in a minute, but before I do that, I want to show you something about ionization of amino acids. This shows the ionization of a simple amino acid. A simple amino acid has two groups that can ionize, an alpha carboxyl and an alpha amine. All right? Now, from your organic chemistry, you should remember the following. And yes, you will need to remember the following. Amines can have a charge of +1 if they have an extra proton, in which case they're an acid. Or they can have a charge of 0 if they've lost that proton, in which case they're a salt. There's the acid form of this amine. There's the salt form of the same amine. Carboxyls, on the other hand, can have a charge of 0 if they have the proton on, and a charge of -1 if the proton is off. The salt form of the carboxyl is the COO-. The acid form of the carboxyl is the COOH. Okay? Let's look at this guy. On the far left in the pink at the top, what do we see? We see that the amino acid has what I describe as all of its possible protons on. It has the carboxyl proton on, and it has the amine proton on. And if we look at that and we add up the charges, we see that the total charge of that amino acid, assuming the R group doesn't have any charge, is a charge of +1. Okay? The middle one has lost a proton, and it's lost a proton off of the carboxyl. And I looked through your comments in the recitations, and one of the comments in the recitations were that I saw that was interesting is, well, how do I know which proton comes off first? How do I know which one loses a proton first? The answer is rooted in the pKa. Remember that the lower the pKa value, the stronger the acid. The higher the pKa value, the weaker the acid, right? Alpha carboxyls typically have a pKa value of about 2.2. Alpha amines have a pKa value typically of about 9.5. You could look at the pKa values and say, and it was a very good question, the one that will come off first is the alpha carboxyl because it's the stronger acid. Okay? The second proton that comes off is the amine, and you see it coming off right here. Now, I want to point out something that frequently is confusing to students. In fact, I've talked to, I think, 2 students already who had this confusion. And the confusion was they associate the pKa with the molecule. A pKa is always associated with a pair of molecules because it's relating to an ionization. To have an ionization, you have one form and you have another. This ionization will occur, all right, in the range of the pKa being about 2.2. The pH around which this ionization will occur will be around the pKa of about 9.5. The pKa is associated with a pair, not with an individual. So we have 3 different molecules, but we only have 2 pKas. Okay? We can think about, for example, acids and salts. All right? The acid of one reaction may be the salt of another. All right? Look at this guy in the middle. All right? The difference between this guy and this guy is what? 1 proton. This guy being the acid, this guy being the salt because it's lost a proton, right? In the next reaction, this guy's the acid, and there's the salt. So whether something is an acid or a salt depends upon what reaction we're talking about. That will help you when you work the problems in the book that have phosphoric acid in them, which has several different ionizations that can happen. Sometimes they're an acid, sometimes it's a salt. It depends on the reaction that you're talking about. Okay. Let's talk about structure. Structure of amino acids. Now, as you look in various books, you will see amino acids grouped slightly differently depending upon the author. Slightly differently depending upon the author. In Stryer, I have adopted the rule that they have for theirs, and we're going to use that configuration that you see here. All right? So if you look at a different book, you may see, oh, well, they don't put glycine in this group. They put glycine in a different group. Don't worry about that. What are you responsible for knowing of what I'm showing you here? All right? Well, you're responsible for knowing the group that any amino acid belongs to. Okay? The group that any amino acid belongs to. So let's go back here. These are the hydrophobics. Okay? Why does it say hydrophobics? That's not hydrophobics. These are the aliphatics. I'm sorry. Aliphatics. Just notice that. So we'll call these the aliphatics. All right? The aliphatic amino acids include glycine, alanine, proline, valine, and leucine. You don't need to know the abbreviations. Okay? So there's one less or several less things for you to have to memorize. You don't have to memorize the abbreviations. But for all of the amino acids, you should know what ionizable groups that they have. These are very simple. They all have only an alpha carboxyl and an alpha amine. They don't have R groups that can ionize. So they only have two, an alpha amine and an alpha carboxyl. Okay? Now I'm going to say a couple things about two of these amino acids in here because they're very interesting. All right? Glycine is the only amino acid that appears in protein that doesn't have asymmetry, meaning it cannot exist in a D and L form. It's the only one that's in that category. And the reason it can't exist is because it doesn't have 4 different things attached to its alpha carbon. It only has 3 because it has 2 hydrogens. Glycine has the smallest R group. The smallest R group. And that R group is a hydrogen. When we talk about proteins and we talk about flexibility, we'll see, and we're going to talk a little bit later about how amino acids can rotate and how their R groups, if they're really big, can get in the way of things. Glycine has the smallest R group. It's the most adaptable, the most flexible for any of the amino acids that we will be concerned with because it has the smallest R group. The other amino acid I want to talk about here is proline. Proline is an odd amino acid in the sense that its R group actually goes up and it comes back and it connects with the alpha amine. It's the only one that does that. Its R group connects with the alpha amine. And you say, well, so what? Well, the so what is interesting because this guy is a 5-membered ring, and because it's a ring, it can't rotate. It's not nearly as flexible as all of the other amino acids are. Very, very little flexibility because of that ring. Now, we'll see that when proline appears in proteins, that we commonly see bends in proteins because of that lack of flexibility. Prolines are commonly associated with bends in the overall protein structure. Okay. All right. Now we move to the hydrophobics. Sorry about that. All right. The hydrophobic amino acids include isoleucine, methionine, tryptophan, and phenylalanine. All right. Each of these is very simple. Each of these only has an alpha carboxyl and an alpha amine that can ionize. None of the R groups in these amino acids can ionize. You can see that some of these guys have pretty bulky R groups. Look at tryptophan. That's a big honker out there. Phenylalanine, also pretty good sized. You notice I said they were hydrophobic, and if you remember from your basic biology and basic chemistry, hydrophobic means doesn't like water. Oil is hydrophobic. It doesn't like water. These guys don't like water very much. Yes, they are water soluble, but they're not as water soluble as the others are. And we'll see this gives them some interesting properties in the overall structure of a protein. We also see that these bulky R groups, to some extent, can play a role in determining protein shape because there's places where they really do get in the way. The only one I'll single out here is methionine. Methionine is one of 2 amino acids that contains sulfur. Okay? It's a bond between sulfur and 2 different carbons. We'll see that the other amino acid that has a sulfur has a sulfhydryl. This is not a sulfhydryl bond here. Okay? Methionine is of note because virtually every protein on the face of the earth, that's the first amino acid in virtually every protein. Okay? We'll talk about that next term, but that's one sidelight for that. This sulfur group that we see in methionine is not, underline not, very reactive. The sulfur that we will see in cysteine is very reactive. Okay? Big difference between those two sulfurs. Okay. Polar. Polar amino acids have groups that can either hydrogen bond or, in some cases, ionize. All right? They include serine, threonine, tyrosine, asparagine, glutamine, and cysteine. There's that sulfhydryl one. Okay? If we look at each of these, we can see, yes, it makes sense that we say they are liking water because they can form hydrogen bonds. Here's a hydroxyl. Here's a hydroxyl in the R group. Here is a hydroxyl in an R group. Here is a carboxamide in an R group. Here's a carboxamide in an R group. And here's a sulfhydryl in an R group. All right? We'll see that that sulfhydryl in the R group can actually lose its proton and become an S-, which makes it very, very much liking water. Okay? As I said earlier, cysteine is an amino acid that has the sulfur. That sulfur is fairly reactive. And most commonly what it will do is it will react with the sulfur on other cysteines. They will join. Very commonly happens. And it makes a covalent bond between them that we call a disulfide bond. And the disulfide bond is very strong because it's a covalent bond. And that disulfide bond can help to stabilize overall protein structure. We'll see some clear examples of that later. But if I put two cysteines close together in a protein, they will form that disulfide bond and help stabilize the structure of that protein. Okay. The last two groups of amino acids all can ionize. The first of those are those that have ionizable amine groups. Sorry. Here. Nope, what did I do? The first of those have ionizable amine groups, and I thought I had... Oh, I've made a mistake in linking. Okay. All right. Well, they are lysine, arginine, histidine. Lysine, arginine, and histidine. Okay. The important thing about them is that each of them has an amine in its R group. And you remember that when amines gain a proton, an extra proton, they have a positive charge. So these guys have three different ionizable groups. It's the first one you've seen, with the exception of cysteine. It has the possible charge of the alpha carboxyl, the alpha amine, and the R group amine. The alpha carboxyl, the alpha amine, and the R group amine. Now, whenever I talk about an ionizable group, whether it's a carboxyl, an alpha carboxyl, or an alpha amine, or an R group amine, or an R group carboxyl, which we'll see in a minute, any of those can ionize. Anything that can ionize will have a pKa if it's a weak acid, and all of these are weak acids. So each of these will have their own pKa value. Each of these will have their own pKa value. Okay? The last two I'll show you are aspartate and glutamate. And aspartate and glutamate look like this, and they have R groups that have a carboxyl. So they have three ionizable groups also. They have an alpha amine, an alpha carboxyl, and an R group carboxyl. And again, each one will have its own pKa value. Okay. Now, you don't need to memorize structure, but you darn sure should know if I say leucine, you should be able to figure out, well, does it have an R group ionizing? No. Does it have an alpha amine? Yes. Does it have an alpha carboxyl? Because everything has an alpha carboxyl and an alpha amine, right? That's simple. So once you know the R group answer, you know the possible charges that that amino acid can have. Yes? Yep. Well, I think both of those would be fair. But yes, you will need to know the names, from the names, whether or not they ionize and how they ionize. Yep. Okay? Okay. So those are the amino acids. Another thing I'll give you is students say, oh, well, do I have to memorize all the pKa values, et cetera, et cetera? And the answer is no, you don't. I will give you enough information to work any problem that you have to work. Okay? I may not give you this exact figure, but I will give you, in fact, a simplified version of this if you need pKa values on an exam. Several of you have asked about old exams, and I thought I had posted an old exam on the web page. I don't see it. So I will post an old exam on the web page so you can see the format of my exams. All right? Use that to understand format. The worst worst mistake students make in my class is they study old exams. Dumb idea. Okay? Don't do that. All right? But use that to understand how my exam might be set up. If you do that, then I think you're much better prepared. All right. So this shows something about the conversion between what they call acid and base, and what would we call that? Acid and salt. Very good. Okay, so acid and salt. And we see the typical pKa value. This table is kind of dumb, and it's not consistent with things. So I don't even tend to use this table. But I will give you enough information with pKa values for you to work problems. Don't worry about that. Okay. Abbreviations. Look at that. All this you're not going to have to memorize. How do you like that? Ah, you're welcome. All right. Let's turn our attention to amino acid ionization again. Okay? Well, I guess we won't do that one. You've already seen in your recitation last week how aspartate can ionize. All right? And I asked you to look at this and in the recitation talk about what does this figure tell you? Okay? So what does this figure tell you? I'm going to tell you what this figure tells me. Okay? Maybe this will help guide you. All right? It tells me, first of all, it's a plot of pH versus OH added. That means it's a titration plot. In a titration, I am changing something in a molecule, and the something that I'm changing is the number of protons in the molecule. I see that as I add more hydroxyl groups, the pH goes up. But it doesn't go up in a linear fashion. It doesn't go up in a linear fashion for the reasons we stated the other day, because the ionization occurs as a result of a log function. Okay? That means, therefore, that when we are near values of pKa, we will see the pKa not going up as fast. And we get away from those values, we see it going up faster. These are close enough together, you don't see it going very fast. But look over here. We get away from there, the value goes up very rapidly. Okay? We see that the flattest places of these plots are where the pKa values are. And that's something that I hope that you took away from last time. These values are what we call maximum buffering capacity. Maximum buffering capacity will occur when pH equals pKa. And they will occur, which also is the same thing, when salt equals acid. We have three different salt-acid forms. One here that you see by this pKa value, one here that you see between this pKa value, and one over here between these two. Every time we have a pKa value, we have a buffering region. We remember that the buffering region is approximately 1 pH unit above or below the pKa value that's there. Okay? That's the message of this figure. Okay. Now, this is an amino acid. I picked an amino acid that had 3 ionizable groups. It has an alpha carboxyl. It has an alpha amine. It has an alpha carboxyl. It has an R group carboxyl, and it has an alpha amine. Notice that the alpha amine was the last one that had the proton come off because it had the highest pKa value. This is about 9.5. This is about 4.4, and that's about 2.2. Okay? All right. So much for the amino acids. Let's get into the good stuff, which are the proteins. Actually, maybe this is a good place for a little break. You guys want a little break? Oh, yeah, we always want a break. Okay, so you know the rules here, right? Instead of me singing this one, I've got somebody to help you out. Okay? So you got to sing it loud. Hopefully. Whoa. Ah. Ah. We're stuck.
Lies in Argentina is basic ones you should not miss.
I can't hear you. All right. Very good, very good, very good. Okay, you guys are doing great so far, so this is a good start. Very good start. All right. Well, with the remaining time we have today, let's turn our attention to beginning to understand primary structure of a protein. The primary structure of a protein, I'll remind you, is the sequence of amino acids that's in a protein. We have 20 different amino acids, and they differ only in the composition of their R groups. They differ only in the composition of their R groups. We can understand, therefore, that the R groups are going to play a very, very important role in determining what the protein function and structure actually are. All right. Here depicts on the screen the synthesis of the beginning, Gesundheit, of the beginning of a protein. It's the joining of two amino acids. The two amino acids are joined in the following way. The alpha carboxyl of one is joined to the alpha amino of another. You're disturbing the class. Turn it off. Okay. The alpha carboxyl of one is joined to the alpha amino of another. All right? That results in a splitting out of water and the formation of a peptide bond. Okay? The splitting out of water and the formation of a peptide bond. The peptide bond is the bond that joins amino acids together in proteins. Okay? This reaction occurs inside of ribosomes. We'll talk about that next term. Okay? And this peptide bond, as we will see, has some very interesting properties that overall determine things about proteins. Okay? One other thing I should point out. When the next amino acid is added, it will be added on this end, and this carboxyl will join to the next alpha amino group. Okay? That's very important because what it means is that when we look at a protein, we can always determine that it has two different ends. One end called the amino end, which is where the only free alpha amino group will be. I say free because it's not bound to anything else. And it will have a carboxyl end, which is where the only free carboxyl, alpha carboxyl, will be found. Okay? The amino end has the only free alpha amino. The carboxyl end has the only free alpha carboxyl. We can describe the structure of a protein as going from amino to carboxyl. And we describe it that way because that's actually the way in which it's made. It's made from amino to carboxyl. We keep adding more and more onto the carboxyl end. That's how we make those. Okay. This shows a peptide that is I won't say a polypeptide, but it's an oligopeptide, meaning that there's several amino acids that have been joined together by peptide bonds. All right? Let's look at what this actually shows us. All right? The first amino acid here is tyrosine. All right? You look at there and you say, okay, there's the alpha carbon. There's the R group. There's the alpha amine. And there was where the alpha carboxyl was, but it joined with the next alpha amine. Alpha carbon, alpha carboxyl, alpha amine, alpha carbon, alpha carboxyl, alpha amine. You start to see a pattern? We had 4 things that were comprising an amino acid, and 3 of them are tied up in the backbone of the protein. The alpha amine, alpha carboxyl, alpha amine, alpha carbon, alpha carboxyl, alpha amine, alpha carbon, alpha carboxyl. The 4th one, which isn't tied up in the backbone of the protein, those are the R groups. Look how the R groups are situated. Do you see any pattern there? Well, here's how they're situated. This one's pointed upwards. This one's pointed downwards. This one's pointed upwards. This one's pointed downwards. This one's pointed upwards. I think you begin to see the pattern. Right? That's how they're arranged. And they're arranged in that way because R groups are big. R groups are big. R groups are bulky, and they can get in the way of each other. We remember when we talked about van der Waals forces, and I said if we try to scrunch atoms too close together, what's going to happen? They're going to have a very strong repulsive energy. It means that if we have big bulky groups, we can't put them right next to each other. There are space limitations. There are energy limitations. And so this arrangement that we see for proteins actually works out pretty well. They're on opposite sides of the same thing. Okay? Even there, we get some steric considerations. Schematically, this now shows you what you saw in the last figure. There's the one R group, there's the other R group, another, etc. Okay. The last thing I will talk about today is something that's a little conceptually hard for students to understand. Okay? So I want to try to help you to understand this. All right? The peptide bond that was formed by joining the alpha amine to the alpha carboxyl, the peptide bond is drawn as a single bond. And we remember from organic chemistry that single bonds can have rotation about them. But it turns out that peptide bonds don't behave that way. Peptide bonds electronically can form a resonance structure that looks like this. And this is completely equivalent to that. And this resonance structure that we see is a double bond. And I hope you remember from your organic chemistry that double bonds cannot have rotation. Now, peptide bonds, because they can exist in a single or a double form, tend not to be rotatable. They tend not to be rotatable. So now if we go back to our original structure that we saw over here, right? We show this as a single bond, single bond, single bond, single bond, single bond all the way through the backbone. But now I've just told you that every time we have a peptide bond, it is not rotatable. The thing isn't as flexible as you thought it was. Every place where there's a double bond, we've got something that is flat. Okay? It exists in a plane. It's flat. It lies there just as it is, and parts of it don't rotate. All right? However, there are things that can rotate. For example, the bonds around the alpha carbon can rotate just fine. This bond can rotate, this bond can rotate, this bond can rotate, and this bond can rotate. Now, if we look at an individual alpha carbon, let's say this alpha carbon right here, and we look on either side of it, we've got two planes, two flat things, one thing over here and one thing over here. In the middle is an alpha carbon. On either side, we've got flat things, right? We can have rotation around the alpha carbons, and I'm going to show you using my nose how that happens, all right? My nose is an alpha carbon, all right? Here are the bonds around that alpha carbon. Here are the planes around that alpha carbon. They can rotate, right? Are you with me? Okay, so they can rotate, and we can imagine that their rotation gives some flexibility, which it does, but my hand isn't really big enough to show the size of the R groups. I'm going to make this extreme so you can see this a little better. Here's the alpha carbon, and here's the attempted rotation. There are some places where rotation will not be favored. Okay? Some places that if we try to rotate, they're going to bang into each other, and the repulsive energy is going to be so strong that that's not going to work. Okay? Now, what that means is that there are limits to the flexibility we have of a protein. Proteins are very flexible, but there are limits to those. A man named Ramachandran, who was a very famous Indian scientist, was the first one to predict this, and he turned out to be right. He showed, or he predicted, that if we look at the rotational angles around the alpha carbon, and he called them phi and psi. Okay, I got it backwards. Phi and psi, sorry. Phi and psi, all right? If we look at those rotational angles and we look at the possible structures, we discover that there are places where things are very stable. They're shown in green. And there are places that are very unstable, and they're shown in white. And he predicted this using a computer. And now using modern methods for analyzing protein structure, Ramachandran turns out to be right. Okay? I'm going to say more about Ramachandran next time. I will let you go until then.